think about what these speakers would be talking about, and obviously different aspects of cell-to-cell signaling related to immune response and others. And so I decided to pick my favorite organ in the brain, but try to concentrate on a strange set of signaling molecules, and those are the D-amino acids, some of which we've been working on a while and some of which are newer. This slide is obviously review for most chemists. When you talk to biologists, it's probably more important, but one of the interesting things, of course, is every biochemistry book that I've ever looked at basically says that, archaebacteria, bacteria, simple organisms obviously use D-amino acids. They're critical components of the bacterial cell wall. But higher animals use L-amino acids, and that's just the way it is. And so protein synthesis is L-amino acids. There are a few exceptions where after you made the protein, you can switch something to a D. But we tend to think of humans, of animals in general, as using L-amino acids. And it turns out that's not always true, and I want to talk about some of those exceptions. It turns out some of the exceptions are actually very important. And if you think about what we think of as our most complex organ, the brain, you would certainly think if higher organisms use L-amino acids, then obviously our brain, being the most complicated organ, must be strictly L, and that's far from true. And so I want to talk about some of these and kind of go through the little bit that's known about these. The one example where we actually know a lot is D-Serine. D-Serine has been studied for almost 20 years, and it actually is known to be made not in neurons but glial cells. It is actually has some functional and actually now disease relevance, and so it's mainly found in the most common cell in the brain, which is the helper cells, the glial, the astrocytes. It turns out one of the most common receptors and the most important, I don't know if I would say common, the most important receptor for learning and memory in our brain and every animal's brain that's really been studied well is the NMDA receptor, the N-methyl D-aspartate receptor. It's kind of funny when you're talking about D-amino acids and you talk about a receptor with a D-amino acid in its name. It's a glutamate receptor. It responds to glutamate. When first being studied, there's lots of different glutamate channels. The physiologist went to an organic chemist and said, give me some molecules that may gum it up, or agonists or co-agonists. And so after trying glutamate and aspartate and D-Glu and D-aspartate, they found, for example, N-methyl D-aspartate also worked. And so that's not considered an endogenous molecule. But it turns out when people study this particular channel in a dish, most of the time they used high levels of glycine. And it turns out this channel only responds if glutamate and glycine, or it turns out, or D-serine is present. So if you have high levels of glycine, then obviously glutamate is the limiting molecule and it works. It turns out, though, that in most cases in the mammalian brain, you have the endogenous ligand for the co-binding site is not glycine, but is D-serine. How this is made is now known. There's an enzyme called serine racemase that actually converts L-serine to D-serine. And it turns out it's oxidized by an enzyme that's been known for a long time, D-amino acid oxidase. What's interesting is before people knew what these enzymes were that made or destroyed in some cases, microarray experiments and other transcriptomics really showed that these proteins that are responsible for making or destroying D-serine, or now we know are responsible, are misregulated, for example, in schizophrenia, and now actually D-serine is actually added to some of the treatments. And so this is now a case where this is known well. One of the reasons I'm telling you this is a story when I actually was approached by some collaborators, or now collaborators, that they wanted to prove that D-serine was released from glia using vesicular release. I thought that was actually a silly question because, as Christy said, that's how modulators are released, through vesicles. And that is, in general, true. However, glia can release a lot of molecules through hemichannels, and so they open up and cytoplasm, in essence, pours out. And there's a lot of articles, there's a lot of papers, and a lot of groups feel that vesicular release of D-serine is not really the major release mechanism, and it's actually caused by this hemichannel, opening and closing of channels in glia cells. And so we wanted to actually start studying that and show how D-serine is released. And so if you think about what I know and others know about how signaling molecules come out of a cell in an activity-dependent manner, this sounds like a silly project. It's kind of like saying, I'm going to show that cells have a cell membrane. But in this particular case, this is actually something that is not currently known. So one way to do that, of course, is to isolate the vesicles that exist in glia and see if they have D-serine. And that's what we've done. And so we've actually taken you can take a section of brain, you can remove the neurons by shaking and other approaches, so you only have astrocytes. And what you can do is you can actually, in this particular case with magnetic beads with a synaptobrevin antibody against, you'll actually grab the vesicles and you can then isolate them and keep them active. Now remember, we want them to be active still so they can still package and release, or I mean still package uptake, for example, and even convert L-Serine to D-Serine. And so this actually has to be done fairly quickly. And you can even go one step further. You can actually heat these vesicles up a little bit, and they start leaking their contents. And so we can see what's in the vesicle and what's in the vesicle after leaking, and even what they can restore. So the goal here is then to show where the D-Serine is. This talk, I decided not to spend a lot of effort on some of our technology development, and so a lot of what I'm going to be showing you are approaches using straightforward approaches such as capillary electrophoresis, in this case with laser-induced fluorescence detection. It's a chiral separation, so we're using a very typical cyclodextrin SDS separation, and this is just showing a typical electropherogram. The glia homogenite, there's L-Serine. You can see that really large D-Serine peak. It's not too obviously obvious here. There's glutamate, there's glycine, there's a little gamma aminobutyric acid, and so these are the neurotransmitters. I guess I have to step back and say the definition of a neurotransmitter in every book that I've ever seen starts out with the first requirement that it's in a neuron. And so when you see GABA or you see glutamate or you even see D-Serine, these aren't neurotransmitters in this case because they're in glia. So some people like to call them glia transmitters. They're the same molecules in some cases, and others they're not. But if you look at the glia homogenate to intact glial vesicles, there's a lot of D-Serine in there. Obviously, these also seem to be found in the vesicles, and you can leak them by temperature heating them, and the contents go way down. The D-Serine almost disappears. So that certainly tends to fit. However, migration time is not enough, and so you have to confirm this in different ways. One way to confirm it, as I mentioned, there was an enzyme called D-amino acid oxidase. It turns out the mammalian form of that is a little bit harder to work with, so we went to another collaborator who actually makes a microbial version where the cofactor is actually covalently bound. It's a little bit smaller, it's more soluble, and it's a lot easier to work with, and it's very active. And so you can take, for example, your sample, and you could add this cofactor, and it'll destroy or eat up, releasing peroxide, almost all of the D-amino acids with the exception of D-aspartate or D-glutamate. So this removes the D-Serine, and since that peak goes away, that's certainly an indication that really is D-Serine. It could be obviously D-alanine or another D-amino acid, but it's not another molecule. You can do further confirmation, and one of the reasons we were doing this is because a lot of our neuroscience collaborators were quite surprised at the rich repertoire of molecules like GABA and D-Serine in these vesicles. And these are actually all from the cortex, I should mention, of rodents, depending on which sample we're using. It's mice or rats. We actually have done several other confirmations. It turns out we do a lot in our group of capillary electrophoresis mass spectrometry. That works quite well. The problem is under chiral separations where we're using SDS, electrospray mass spectrometry isn't too easy to do in the presence of surfactants. And so we used a non-chiral approach, and that's shown here. We quantified that. We actually also then verified with a non-chiral under different conditions with CE-LAF. And what's really kind of nice is everything actually matches. And so leaked vesicles have no D-Serine. Intact vesicles, as I showed you, have quite a bit. L-Serine is there. The totals are here. And look at the non-chiral, give you a similar total, whether leaked or not. And even the CEMS corroborates this. And so at this particular point, I'm willing to say that we are measuring D-Serine in the vesicles. It leaks out. We can actually restore it by waiting a while. We've also done non-chiral and actually verified, for example, the GABA, the glutamate, and the other molecules. And so astrocytes have a rich repertoire of cell-to-cell signaling molecules that if you just took a section of brain, you might call neurotransmitters. One thing to point out is, if you go to Wikipedia, I don't know, I use Wikipedia in this case because every single text book seems to give different numbers, but a microliter of a mammalian brain supposedly has about 100,000 neurons and about 400,000 glia. Depends on the brain region. And so again, there's a lot more glia astrocytes than there are neurons. If you go to an older book, it'll say it's 10 to 1. And so there's actually a lot of the glutamate, and even now the D-Serine seems to come from astrocytes. One of the reasons this is important, actually, if you look at our original work that I just showed you, I mean, the GABA peak actually doesn't seem to be, or is not all mostly GABA. It's actually a co-eluting compound, and you can see that by changing the conditions. There's almost no GABA in this particular set, and even by CEMS. There you don't need a chiral separation. So it really is important to use these multiple approaches. If you go to synaptic vesicles in the cortex, it turns out there's almost no, at least we don't measure very much D-Serine, and the reason, we're actually making a difference here between neuronal vesicles or synaptic vesicles and astrocytic vesicles. And so they are different compounds. Obviously, glycine, glutamate are found in all of them. If you go to the enteric nervous system of a mammal, obviously this has been tested in some, such as rodents and humans, not all, but I'm just using a general case. Actually, D-Serine can be found in neurons, actually in some spinal neurons, and so D-Serine is a glia transmitter. There are exceptions, and so depending on where you're located, it could be a neurotransmitter also, but at least here, it seems to be strictly glia transmission. So to summarize this in terms of the CE work, there's actually a lot of D-Serine in astrocytic vesicles, and there's also obviously a lot of glutamate, which was known. There's L-Serine in synaptic vesicles and very little D-Serine. And so it's where it should be if it's to be released. And so this actually seems to fit with the idea that D-Serine is released. This is a collaboration of about 6 different groups, actually in several continents. And I actually just talked about some of the work that we're doing. You can actually use immunostaining and co-localize the enzyme for serine racemase, and it's in the vesicle membrane. And so the vesicles that we're isolating actually have the racemase. The EM shows that their vesicles are actually the size and shape you would need for release. Radiolabeling assays show that it's, this transport is driven by a proton gradient like most others. And actually, we've gone so far as to, you can actually add that same enzyme, D-amino acid oxidase, and turn on or off D-Serine signaling. It can actually make it into the synaptic or the glial cleft, and you can actually see what happens when you turn it off. And so all of this fits with a role of serine being released from glial vesicles. And so just like neurons have their own vesicles, glia does have its glia vesicle, and it seems to be a proton pump, ATP dependent. What we don't know yet is we haven't, we're taking collections of cells and collections of vesicles, and so I can't tell you what's, D-Serine vesicles also have glutamate or GABA, or there's subpopulations, but at least within astrocytes there certainly is a serine vesicle population. So that's a story that's well known in terms of pharmacology, in terms of mammalian biology, but there's many other D-amino acids found throughout your body, or at least several other key ones. Aspartate, glutamate, and alanine, and I just wanted to talk about a few of those. We've been working on D-aspartate for quite a while. An important part of D-aspartate is found in every animal studied, and I'm not talking about mammals. I'm talking about anything from ctenophores, comb jellies and jellyfish, all the way up to slugs, insects, as well as humans. And its function is not well known, and so I'm going to talk about that. That's some studies that coming up next. There's also glutamate. This is some much more recent data from another group, and even there's D-alanine found in this case in the same cells that release insulin, the beta cells. And so there are other cases, and I'm just going to talk about a few of these. So my group has really spent 5 years trying to say or look at and determine if D-aspartate is a neurotransmitter. As I mentioned, D-aspartate is found in almost every animal studied. It's found during embryogenesis, that's during the formation. And actually, at some point during embryogenesis, some fraction upwards of half of the aspartate, the free aspartate, is actually converted to a D form. It does something that is not well understood, and then it disappears except in parts of the brain, the testes, the pineal gland, and a few other areas. Since it's found in every animal, we decided to pick the animal that is easiest to study the functions of neurotransmitters. The problem is people have actually shown that you can take cells and add D-aspartate and see peptide release. You can take other cells and you can actually see aspartate is converted from L to D, but it's never in the same cell. And to show molecules are neurotransmitters, you have to basically show that a cell can make the molecule, transport it to a release site, release it, and then look at its postsynaptic cell, and you have to do that in a defined way. And so we're really trying to do those types of experiments. And we do what a lot of biologists do, we cheat. So we work with a sea slug, Aplysia californica. Model systems like mollusks have been used to discover many fundamental neuroscience questions, even things like synaptic transmission and even the Nernst potential using the squid giant axon. One reason is some of the cells are giant. This is really large. We're working with a range of cell sizes. And so you can actually then start actually probing these. The other aspect is every animal has these cells. And so you can actually go into the same cell every time and ask interesting questions. When we started this, actually, there's no D-aspartate race mace known in any animal. We and one other group just reported one last year, but since you don't know the enzymes that are making them, you don't know how it's being destroyed, and the enzymes don't exist, you really have to just take apart the brain and analyze it. And we found one group of cells, or actually several groups of cells, that have ridiculously large amounts of D-aspartate. Other cells have none, muscle cells have none, and so the F and C and the G cluster, up to 70 to 80% of their aspartate is in the D form. So these are likely to be important cells. These are neurons that have different functions, in some case known, in some case not known. So we concentrated, for example, on the F cluster because we knew something about that group of cells. You can go much smaller. Those are larger cells. This is the size of some of the neurons in your body. Like a Purkinje cell, it's 30 microns in diameter. Here's a cell soma. Here's a process. If you're careful, if you're not like me where I drink coffee, you can actually dissect out processes, for example, versus the cell soma, and you can see that this cell has quite a bit of aspartate versus L, both in the process and the soma. So the D-aspartate is found everywhere throughout this cell. That certainly is important if it's a neurotransmitter. You can actually determine that the transport is in a vesicular manner, in a colchicine-dependent manner, because colchicine blocks vesicular transport. So we actually took several abdominal ganglia. You can actually cut it off, take a section just to see how much of L or D-aspartate is there, and then you actually wait 24 hours, or you wait after you've added colchicine, and you look if you're accumulating vesicles, because what is known is that under normal conditions, the vesicles will accumulate at the cut end, so they're being transported towards a release site. And it's a kind of complicated slide, but we're actually, when you incubate without colchicine, you can see that both D-aspartate and L-aspartate accumulate at the cut, and this is just the control. And when you add colchicine, there's no accumulation. It actually decreases slightly. So there is accumulation at the cut. We're working with a sea slug. I've been told by many people it's probably because they eat seaweed, and who knows what they have in it. Maybe you're just feeding them D-aspartate. So we used a very retro experiment. We used radioactive aspartate. We fed the animals, not the animals, but the cells in a dish under media, radioactive L-aspartate, and 40% of the aspartate after that in a ganglia are now in the D form. If you actually heat the cells up to 50 degrees for just several minutes, you get no conversion. It does seem to be enzymatic, and in other areas you don't get much. Interestingly enough, if you feed it radioactive D-aspartate, you get almost no back conversion into L. So this conversion seems to be one way, and it's location dependent and different brain region dependent. And this is just looking at the sheath where there's a lot of terminals versus the cell soma. The D-aspartate is uptake in this particular case in individual cells. We're still using radioactivity. It's a nice quantitative approach, and different cells can take up different amounts of L or D. And it turns out it's a sodium-dependent, temperature-dependent manner, which is what you would expect from most amino acid transporters. And so these cells can make aspartate, it can transport it to a terminal, and it can uptake D-aspartate. And so all of this is certainly becoming consistent. Does it release D-aspartate? A lot of things in a cell aren't released in an activity-dependent manner. This is actually a nerve with, looks like chicken pox, but what it is these are about 30 to 50 micron solid phase extraction particles, basically an HPLC column, an old, with the large particles that we spread on there. We can stimulate release using potassium or calcium ionophore, for example, ionomycin, and you can see release. In this case, I'm showing you that there's some D and L-Asp before we stimulate release, The neurons are down here, but we're stimulating down here, and you can see that when you stimulate with liatomycin, we get so much D-aspartate, we overwhelm, and then we can spike and show really what it is. Very few compounds are released, so the electropherograms are simple. You can induce D-aspartate from an entire ganglion. This shows you one of the problems with working with these animals. Most people who work with rodents tend to think about they're using, for example, genetically similar, if not identical animals that are reared in laboratory environment. These are wild-caught animals. We don't know anything about them in terms of their history. They're shipped to Illinois, and then we do experiments. In this particular case, you can see D and L-aspartate, and what we've done is we've actually stimulated. This is from an entire cerebral ganglion, and then we spike with a standard, and we can see actually in a potassium-dependent manner we get an increase. What's interesting, if you measure D and L-aspartate from animal to animal, some animals have only L-aspartate release, some are almost all D-aspartate, and they vary. And so there's a lot of variability. And interestingly enough, this is an experiment that you can't keep on repeating, and so you get one try for each animal when you're looking at this. But there's a lot of variability, and this may deal with animal state. It may deal, it's not, I would say, uncertainty in the normal sense, is we just don't know the physiological state of the animal before starting. Now, what's interesting is that because of the way physiologists measured glutamate channels in the 1970s, almost all the different glutamate channels are known to respond to glutamate differently than aspartate, differently to D-Glu, differently than D-aspartate. This is just a way of categorizing channels, and so this was known in advance. But we, again, wanted to measure in different cells, for example, the R2 cell, which has D-aspartate, we wanted to validate that D-aspartate has a different effect. This is a stimulation of D-aspartate. We see action potentials, and L-aspartate doesn't. We reverse the order here. L-aspartate, you get a few spikes, D-aspartate a lot. And so there are differences in the response. Interestingly enough, if you want, D-aspartate acts as a hormone. If you dump on D-aspartate into the basically the blood, the hemolymph, the R3 through 13 cells release peptide. And so I'm not going into the mass spec peptide detection, but this is a mass spectra looking at the release of peptides from one cluster of about 12 neurons, and D-aspartate can cause peptide release. So that basically says it's acting as a hormone. You can go to the single cell level and back to physiology, and you can dump on either on the cell soma or in a terminal, and you can see that both L-aspartate and D-aspartate cause either, depending on the cell, a hyperpolarizing or depolarizing effect. And so they both, the channels respond to L and D-aspartate, but this certainly indicates that some of the channels do respond to D-aspartate. So what we know so far is certainly everything fits, but one of the biggest problems, we didn't know what enzyme made it. And so we actually spent several years, and earlier, actually last summer, we finally actually characterized the enzyme D-aspartate racemase. And all racemases that are known have dehydratase activity, so this was actually our initial assay, but this can convert, this is L-aspartate to D-aspartate. The serine racemase, for example, has a pretty slow conversion efficiency too, so this isn't too surprising. This is a useless slide to all of you, but if you show enzymes, people like to do this. It's, there's the gene. It's important to realize that, so this is the first invertebrate D-aspartate, neuronal D-aspartate known, and actually about a month before we published this, Saul Snyder published a very different looking enzyme, which is the first mammalian aspartate racemase. And so there's two known. His actually uses a very different mechanism, and it has a very slow conversion. And so, even though genomes are known, some proteins that are either unannotated or misannotated seem to have things like aspartate activity. All known racemases actually that are found in animals require the cofactor PLP, and so does this one. And so it does, it's a good sign that it seems to have something similar to most other known racemases. And just to show you, we did characterize this enzyme, and it does what you would expect. The most interesting thing about this enzyme is, so it actually has a pH dependence that's optimized for Aplysia pH, a little bit different. It requires ATP, it's increased magnesium. All of this seems to fit. The last thing that's interesting about this enzyme is it actually has the ability to make both aspartate and serine, and so it actually has a dual activity. And so it's one of the first racemases that's been characterized and reported to have the ability to convert both L-serine to D-serine and L-aspartate to D-aspartate. And so that actually is somewhat unique. We wanted to verify that, and I'll show you, our endogenous measures. We never actually looked for D-Serine in Aplysia before. The other thing that was kind of fun about this is that we had taken apart the Aplysia CNS, and a very dedicated graduate student had made many measurements with capillary electrophoresis, LAF, and found that the F and C clusters had the highest levels of D-aspartate, and compared to the abdominal ganglia, the pleural, the pedal, and so when we had the enzyme, we can now do in situ, and we can see that the F and C and clusters actually, for example, light up, and there's lots of staining there. And this is just showing the buccal ganglia has a little bit, some processes in pleural and pedal. And so we had identified the areas of the brain that had the highest amounts of this enzyme, and those are the same area that had the highest amounts of D-aspartate. So the fact that we did this backwards and did D-aspartate first and then found the enzyme is interesting. It turns out we can change the conditions, and there's actually lots of serine, D-serine in the same areas as there's aspartate. So it does seem to be the same enzyme that makes both. So everything fits the same, with D-aspartate does fit the criteria of a neurotransmitter in the C cluster of Aplysia. It may also fit in mammals. It just is very hard to do these types of studies. Snyder's work shows that it has a trophic factor, and it actually changes survivability of neurons. But just like serotonin, a lot of molecules can do multiple roles. So I want to go and end with some work we're doing on glutamate. Glutamate is actually, D-glutamate is a funny analytical story in the sense that you can take the hippocampus of a rat, and you'll see reports that 0.5% of the glutamate is in the D form, so it's not very important, right? I mean, who cares about 0.5%? Well, this is some data, and there's some others. There's now D-glutamate antibodies, and they show very sparse staining. 1 in 100, 1 in 200 neurons have all the D-glu. So now it's changed. Rather than neurons having 0.5% of D-glu, now it's 1 in 200 neurons have all of it. And now it becomes the question, what are they doing? And so this is in the different locations, the Rafe and others, but you can see this. And so we wanted to see if we can measure D-glu. It's a little bit hard. We can actually measure some D-glu, but it's, and you can do a very nice separation to get D-glu. But the problem is that's, it's a very low peak. So we tried D-aspartate oxidase digestion. It's a different enzyme, which also digests D-glu. And we did it to 1 to a, so now we have low levels of D-aspartate, nanomolar, compared to the others. And it turns out at this level, the enzyme doesn't work well. So we went back to the drawing board. Obviously, if you do this, you get rid of it, and then you don't detect it. And so this enzyme gets rid of the signal. So we re-optimized quite a bit and actually used a slightly different form of D-aspartate oxidase enzyme, really to make it optimum for D-glu. And it's starting to work. This is just showing you that in a homogenate, you can get rid of D-glu down to almost nothing. The aspartate also goes away, and so now we can start confirming. Going back to some of the sensory neurons and some of the other clusters in the brain where we had both aspartate and maybe D-glu. And you can see we have, for example, a small D-glu peak, and you can remove it by this D-aspartate oxidase. It's gone. And so this does indicate that we are detecting small amounts of D-glu. The next step, of course, would be to isolate the particular cells, and that actually is a little hard because, again, there tend to be, we expect them to be rare cells. And so aspartate is common, 30 to 40%, up to 80% in some brain regions of Aplysia. The D-glu is 0.1% or several tenths of a percent are very low. We have measured release. So if you stimulate with potassium, you can actually see a peak that seems to match the D-glu, and so this is before stimulation. After stimulation, the peak rises, and so whatever this peak is does go up. We presume by migration time it's D-glu. And last data slide is simply saying we actually have tried this on multiple sensory neurons. Most we detect none. Here's one of the few cases where we actually detected a fair amount of D-glu in one cell, and we spiked, and it does seem to match. This is a different set of separation conditions, and again, in the L8 neuron, where the sensory neurons form synapses, D-glu gives you a bigger response than L-glu. I will say I actually went to a pain meeting, or the American Pain Society meeting, for a very different purpose, and I discovered that there was many groups that calibrate L-glu release in the spine by feeding the cells radioactive D-glu. The cells take it up, and they subsume in the same synaptic vesicle and release it. And they said, since D-glu is biologically inactive, this is a great control. Well, the answer is maybe D-glu really isn't biologically inactive, and their 20 years of control experiments may need to be looked at again. So D-amino acids in the brain. They're there in every animal looked at. D-Serine is important for learning and memory, potentially schizophrenia, or actually, I guess, by treatment it is. D-aspartate in Aplysia fits the role of a classical neurotransmitter. It's found in neurons in vertebrates. And D-Glu is really not well understood. It's in sparse networks, making it hard to study. I will add that the other point is there is no non-microbial, non-plant model system where a D-Glu racemase is known. That's not to say it's not there. There's actually lots and lots of enzymes that haven't been well characterized. There's lots of serine dehydratases that haven't been tested. We know, it seems to be made, but one of the problems is we can't find these cells using enzymology, looking for the racemase yet, because this has not been identified. And just to, as teasers, D-alanine and D-pro are the last two amino acids found at high levels. And again, this is some really fun work by Hamas and others in Kyushu University in Japan, and he basically shows that D-alanine is found in the same cells that contain insulin and not in, so this is an islet, and basically his comment is that there's very high levels of D-alanine in the beta cells, and so you think about all the work that's gone on in diabetes and insulin, and to find a D-amino acid in some of those cells that isn't well characterized is a surprise. One of the reasons these haven't been well studied really has been the lack of analytical techniques and also the overwhelming knowledge that, of course, that D-amino acids aren't in these cells. Why would you look for them? I think has held back this field for a long time. And so probably in the future, you'll basically it'll be well accepted, or we'll understand a little bit more of the roles of proline, alanine, aspartate, glutamate, and serine in their D forms in cell-to-cell communication. And lastly, I thought I would end with a slide of the group. As you can see, this was taken on April Fool's Day last year, and so the group has changed a little bit, but I thought it was appropriate. D-amino acids do seem to be responsible for hair color. And with that, I would be very happy to either take questions, or I guess we can release everybody for lunch. Your choice. Thank you. Well, so in the case of D-Serine, obviously so. It's the NMDA receptor. And NMDA receptors require glutamate and D-Serine or glycine to be activated. So in many cases where there's glycine level is not high enough, then if you want to do learning and memory through an NMDA receptor, if you want to activate that in any pathway, you have to have D-Serine. So the D-Serine signal from an astrocyte probably is much longer, right? So you activate the astrocytes, D-Serine is there at a longer time constant, and then you get the glutamate signaling, which is fast, and that's how you activate those. Aspartate activates some known glutamate channels. There's some low affinity glutamate channels that may react more to D-aspartate. But that's not as well understood. And when you're starting to talk about others like D-glutamate or D-proline, D-alanine, nothing is really known. So the answer is there is physiological evidence on how D-Glu and D-Asp respond to glutamate channels, and it's presumed that they're fitting into the same binding pocket, but there's been less studied. I've had people ask me that before. So, again, in D-Serine case, that's certainly not true. In the case of aspartate, for example, if you think about what we know about it, it's found in subsets of cells only in the pineal gland, the testes, and a few select neurons. It becomes, a little bit weird to say, why do these cells do this? It releases them in the D form in an activity-dependent manner. It seems to be in vesicles, and so that becomes a little bit strange to call it a metabolic reservoir. If you think of the data just during embryogenesis, that there's only select time points that you actually find it, again, it's not to say it couldn't be. In the case of D-Glu, it's again, whatever you want to say about it, it could be, since nothing is known, it could be a metabolic reservoir, but the strange part then is why does only 1 in 100 cells need that reservoir? What are the other 99 glutamate-using neurons doing? So there's something about those cells that are different. But again, if in the cases where we don't know what, I guess you could speculate a lot of uses. My thought on some of these, since the release, is they have something to do with signaling. And again, that's speculation, except in the case of the D-Serine, and in one sense, the functional tests we've done in the D-aspartate. So there's two types of enzymes. There's those that make it, the serine racemase, the aspartate racemase. In the case of the serine racemase, that's located in the glial vesicles. The D-amino acid oxidase is extracellular, and depending on which model organism, it's actually either soluble or membrane bound, and I don't remember what cell type had it. In the case of D-aspartate oxidase, that's also membrane bound. I don't know. Snyder's brand new D-aspartate racemase, I don't remember where that was located. I know it was in terminals, but I don't remember if it was membrane bound or cytoplasmic. Most of that was NDA.